This is 5 Minute Science, and welcome to Level 3, everyone. Well, I probably shouldn't say everyone, because I got a feeling most people got sick of my rambling halfway through Level 1 and want to do something else. But, who needs fun when we have science, am I right? No? That one's just me? Yeah, that seems about right. Anyway, to the three of you who've stuck with me so far, I hope you're ready for some sciencey goodness, because here comes the payoff. Are you ready? Bats! That was a fantastic non sequitur to get me into the first topic I wanted to hit on, zoonosis. Remember back on Level 1, when I talked about how Ebola wasn't a very good virus? Well, that's only partially true. It's not a very good human virus, but it does perfectly fine in animals, like our good friend the fruit bat. Zoonosis is the process during which a virus spreads from animals to humans. It's something I'm sure you're all familiar with, even if it's only by example. Swine flu! Sorry, something in my throat. I'm good. So when humans come into contact with the bodily fluids of an infected animal, which can also include chimpanzees and gorillas, the virus can move into its new host and a human outbreak can begin. But what does the virus actually do to the human? In the last video, I rattled off a list of symptoms, but now we're going to take a closer look at why they actually occur. This is equal parts fascinating and terrifying, so if you haven't watched my first two videos on why you don't need to be afraid of Ebola, I'm talking to you people who only watched the last 3 episodes of a TV series just so you could see how it ends. Go watch them. Here's an annotation so you don't have an excuse. Ebola is a filovirus, which is a strand of genetic material, in this case RNA, covered by tiny filament proteins. Those proteins both protect the virus and pull open the membrane of any cells it wants to infect. In Ebola's case, the cells they target first are part of the immune system. The infected cells release proteins that cause tiny clots to form in the bloodstream, as well as causing widespread inflammation. These clots damage red blood cells, which carry oxygen throughout the body, and create leaks in blood vessels. Eventually, the virus reaches the liver and destroys the cells there, at which point blood loses its ability to clot altogether. This causes blood to pour through the tears in the already damaged blood vessels. Death from severe drops in blood pressure and multi-organ failure usually occur within 6 to 16 days after symptoms first appear. Equal parts impressive and terrifying. Let's lighten the mood for a second with a picture of a chinchilla who won't turn your blood vessels into Swiss cheese. Okay, so now that we know how Ebola works, how do we stop it? At the moment, there really isn't a definitive answer. The main form of treatment right now is just to maintain fluids and throw drugs at the patient until something works. And while this can be effective, it doesn't really give us a clear picture on what's working and what's not. However, there are a couple frontrunners in terms of specialized treatment options. The first is using blood transplants from Ebola survivors in the hopes that their antibodies will help boost the immune system of the patient. And a quick tangent while we're talking about Ebola survivors, there's a study out of Hamilton, Montana that suggests the severity of Ebola symptoms could be related to genetics. In a study done on infected mice, they found that mice whose immune systems responded to the virus with increased cell death and widespread inflammation had worse symptoms and a higher death rate than mice whose immune systems responded by stimulating blood vessel repair and increasing production of immune cells to fight the virus. Now, I know what you're thinking. Of course it makes sense that inflammation made symptoms worse. Ebola is a hemorrhaging virus and increased blood flow to an area would only increase the blood loss. That's basically common sense. And you're absolutely right. The main reason I brought up this study is that within our lifetimes, it's entirely possible we'll be able to control the activation of these genes, essentially enabling the body to heal itself in the most efficient way possible. Okay, tangent's over, but the main point is genetics is awesome. Back to Ebola treatments. There are also 2 drugs being researched to fight Ebola, and I'm going to butcher both of their names. The first is brincidofovir, which is odd because it usually works against double-stranded DNA viruses, and Ebola is a single-stranded RNA virus, but if it works. The second drug is favipiravir, which is believed to impact the effectiveness of RNA-dependent RNA polymerases. In normal English terms, both drugs basically stop Ebola from replicating itself. The last thing I'm going to talk about, and I promise this actually is the last thing, is Ebola vaccination. Now, normally the vaccination process takes years to get out of development, but the severity of the Ebola outbreak in West Africa has led GlaxoSmithKline and the Public Health Agency of Canada to push to have 20,000 doses ready by early next year. The vaccine they're working on took an Ebola gene and put it in an entirely different virus, which means anyone who gets the shot cannot get Ebola from the vaccine itself. Okay. I think I'm done now. Yeah, I'm good. Was it as good for you as it was for me? Probably not, but I am a guy, so that's pretty par for the course. Am I right, ladies? Going out on a high note with a sexual innuendo. I should probably be ashamed of that, but I'm surprisingly okay with it. Thank you all so much for watching all the way through this series on Ebola, and I hope you learned something along the way. If you liked what you saw, be sure to like and subscribe. On behalf of the 5 Minute Science team, thanks for sticking with us, and remember, it's just science.